Hi, and welcome to the Precision Medicine virtual event. My name is Seth Crosby, and I am at Washington University. Specifically, I'm at the McDonnell Genome Institute there. I'm going to be talking about rare alleles. Specifically, the title of my talk is Tissue and cfDNA Rare Alleles, How Low Can We Go? Please feel free to submit questions during my presentation, or you can email me, and I'll follow up with you if I can. Okay, we're on my first slide, and I just wanted to say that we're now actually an amalgam of two different facilities. We're the one that I started, Genome Technology Access Center, and the August McDonnell Genome Institute. We're combined into a single facility. Here's my email address. You are most welcome to contact me. Just a quick description of our facility. Most of the work we do is research, but we do some clinical work as well. We're CAP/CLIA certified. We have even the New York State certification. We do a lot of different genomic tests. And again, if that's of interest to you, that's great. It's just the talk I'm going to be talking about today is relatively clinically based, so I just wanted to mention this. Okay, so a few years ago, we decided to, at GTAC, we decided to start a clinical sequencing facility. And one of the first panels we used was a cancer panel. We brought this panel online about the same time that Foundation got started with theirs. And as you can see, ours had about 20 odd genes in it, while Foundation's had 400. We did this because we wanted to have a panel that was covered by CMS and by various payers. And so we started off with a very modest panel. It was a biotinylated bait panel, not an amplicon panel. And it allowed for very deep sequencing. This is the current state of the panel. You could see that it's grown to some degree, and I'd be happy to send you more information on that. So I mentioned to you that that the panel allows for deep sequencing, and the question is, why do we need deep sequencing specifically in the situation with oncology? The problem is that, as I mentioned to you, a lot of times oncology DNA, malignant DNA, is present at a relatively minor amount in samples. So you'll have, for example, you can see here, there's a whole bunch of, if each of these lines represents a sample, you can see that there's a couple of Gs that are sticking out in the middle of there. And those Gs might be clinically significant, or they may actually be sequencing errors. The sequencers are not perfect, and every now and then, either by PCR or the sequencing itself, an error can be introduced. And the question we're going to be talking about today is, how do we distinguish between true mutations and sequencing errors? This is from a paper that came out a few years ago where they were actually looking at tumor tissue, and they determined that there were a number of different mutations present in different physical areas in this renal sarcoma. And actually the majority of the tumors were not universal across the entire tumor. So this is sort of reminds us that tumors can be polyclonal, and sometimes the clone of interest may not be the main clone. One of the authors said something kind of funny. He said, "I'm still depressed about it. If I'm honest, if we had higher resolution assays, the complexity would be far worse." So tumors are complicated tissue. In addition, the pathologists in the audience will recognize the fact that the section we're looking at is mostly normal, that the tumor really is only the shaded part, and the rest of it is just fibrotic tissue. So in addition to the fact that a tumor can be polyclonal, even a monoclonal tumor, the material of interest may only be the minor material in the section. Okay, so when you come out with any kind of a clinical test, even a lab developed test, the first thing you need to do is to test it, to validate it, to make sure that it works properly, and more importantly, when you validate it, you want to figure out what the sensitivity is. How low can you go? If I talked to you about the complicated situation with cancer, how far down can you go in terms of sensitivity, in terms of the ability to detect mutations? So we came out with this paper, and I'm just going to show you a single image from the paper, just one of the many facets that we tested when we were testing this new panel. And we did a very simple test, is that we just took two defined DNA samples called HapMap samples. These are DNA samples which have been rigorously tested, and we mixed them together. So we took a lot of one and a little bit of another, and we mixed them together to see how far down we could go to detect the allele in the minor population of what we mixed. So we mixed it 50-50, 20-80, 10-90, et cetera. In addition to the physical mixing, we also did some downsampling. We did mixing in silico to further the sequencing test. And this is one of the figures that we did. So this was looking at a heterozygote allele. So we mixed 50-50, but you can see that the top number there is 25% because it was a het. And what on the x-axis, you can see what the coverage was. And I told you that original panel allowed for 1,000x coverage, but we went ahead and downsampled from 1,200 down to 100 coverage. And you can see that at a 25%, that we were able to detect that allele down to about 200-fold coverage. When you drop to 10%, the data still looks pretty good. Even at 900, 800, 700 fold coverage, we were able to detect that minor allele. As soon as we dropped down to 5%, however, we lost the ability to detect the minor allele. And so just simplistically speaking, the story is a little bit more complex, but simplistically speaking, we said, okay, we've got a clinical test, and we're able to detect a minor allele down to 10%. Anything less than 10%, we probably won't be able to call that. So this was using an aligner that was available at the time. But what we wanted to do while we were doing this is we wanted to look at other methods for detecting rare variants. This was another paper that came out actually, I believe, at exactly the same time. And David and Eric were testing other variant detectors. GTAC is the one that we were using for this clinical test. But they decided to compare this with SAM tools, and then a couple of detectors that were created to detect rare alleles. So, for example, Splinter, you actually use spike in DNA in order to be able to define what the error rate is, and Verscan 2 also uses base quality filters to be able to detect a more rare allele. These were, and I know that Splinter was, and I think Verscan was as well. They were used back when sequencing was kind of expensive and people liked to pool samples together. And so they were still looking for constitutive alleles, but because they were pooled together, they weren't there on a 50-50 level. Okay, so again, the paper is a little bit more extensive than what I'm going to show you, but what we did was we basically took 5 HapMap samples, and we identified from our cancer panel 427 heterozygotes that were common across those 5 HapMap samples. And we used the variants had to be detected by both of our standard variant detecting tools, GTAC and SAM tools, I'm sorry, GATK and SAM tools, at greater than 50% detection rate. And so a little bit like the other experiment that I described, we took those 2 HapMap samples and we mixed them together at various concentration. We used our panel to enrich for the regions that we were targeting, those oncology genes. And we sequenced map by Novaline and then used the various detection tools that I just mentioned and compared them. So here are the 5 HapMap samples that we used. And what we did was this was sort of our soda sample right here, 989, and here's the 4 scotches that we mixed with it. And we mixed it, again, as I said, half and half, 20%, et cetera. Now, and looking at those heterozygotes, those heads now, for a half and half, the heads would be present at 25, 10, 5, et cetera, et cetera. So the first thing we did was we looked to try and see how many false negatives we have. So here are the 4 tools again. These are the standard tools, GTAC and SAM tools. And then here are the more sensitive tools, VeriScan and Splinter. And here on the x-axis, you see we're diluting our allele of interest out from 25 to 2.5%. And the first thing you notice is that the standard allele detector, which was built to detect either a homozygote or a heterozygote, doesn't do a particularly good job detecting a rare allele, even at 25%. SAM tool starts to degrade a little bit, and then this gap right here, where you see zeros, that's SAM tools zeroing out. It's no longer able to detect that variant. And that's not surprising because it was not designed to detect it. GTAC does a better job. It's able to detect to some degree down to 5%, but it zeroes out again at 2.5%. Whereas the variant detection tools that were designed to detect low frequency alleles did a pretty good job. Looking at specificity, now we're going to look at false positives. And as our teachers in college told us, what you give up in sensitivity, you gain in specificity. And what you can see is that we're measuring false positives on the y-axis. And you can see that GTAC and SAM tools had a relatively small number of false positives. As a matter of fact, look at it like where SAM tools are. You can see those zeros again on the x-axis, indicating that there were no false positives from SAM tools all the way down to 5% or 2.5% heterozygote presence. Whereas VeriSeq and Splinter, you can actually see, interestingly, that Splinter's number of false positives increased as the allele frequency went down, and VeriScan was relatively constant. So if you just look here sort of as a kind of a 6 mile up comparison, the green indicates good, the yellow not so hot, and red is not great at all. We could say that Splinter sort of takes the award because it was very sensitive, as it's designed to be, and it wasn't too bad in terms of specificity. You can see here that Splinter, even at 5%, was still not a lot of false positives. And I put a little silver medal there next to Splinter. But I want to remind you that these tests were run a few years ago, and they are constantly reversioning these allele testers. And you really need to repeat this experiment with current data to be able to determine whether or not what your sensitivity is currently. All right, so what we've been talking about up until now is detecting DNA derived from solid tumors. Now we're going to go over to cell-free DNA. In the case of oncology, it's also called ctDNA, or circulating tumor DNA. This DNA was first, circulating free DNA was first noted in plasma in 1948. It was noted to be elevated in cancer patients in 1977. And Vogelstein started looking at those mutations and being able to detect rare mutations using a method called BEAM in 2007. Circulating tumor DNA doesn't hang around in plasma very long. It's only there for less than an hour. And the presence of the allele can be very low. Just basically put a question mark, and can be as high as 1 out of every 100. It could even be higher than that, I've been told. And so here what you have is just some vessels that are running through a tumor, and the tumor cells, are necrosing and releasing DNA into circulation. And this is thought to be, at least in the case of solid tumors, how circulating free DNA is released. And this can be a primary tumor, or it can be a very small MET. But this is a way to detect the presence of tumor without actually having to biopsy. So, as I said, the minor alleles in serum or in plasma can be very minor indeed, and the problem remains is how do you detect an allele that's extremely minor? One nice thing, for example, if you had identified an allele in your tumor, and you could look for the presence of an allele that you'd previously identified in the tumor, or you could look at common mutations, medically actionable mutations that have already been identified and are often present in about 85% of malignancy. So it would be nice, for example, you could design a PCR analysis where you could see here that you have two templates here, a germline template and a mutant template, and the primer sets down on the DNA in the presence of the mutant template, but it doesn't match in the presence of germline. So great, you just run a PCR analysis, and if you get a product, then you know the mutant is present in the plasma. The problem is that the PCR tends to be it tends to be a little bit leaky. In other words, somehow the polymerase is able to get around that non-annealing base. And so, for example, this is some data that was generated at IDT, one of our companies that we work with, and the company I'm going to be talking about that invented the next technology that I'm going to describe. And you can see in these two tables, this is RT-PCR, this is quantitative PCR, and what you can see is that the number of cycles on the x-axis indicates how much material is present, and so that the more material that's present, the fewer cycles are required in order to make a product. And so in this case of a mismatch, C to A mismatch and a T to G mismatch on the left and the right, you can see that there really is just about 1 or 2 cycles difference between the wild type and the mismatch, meaning that the TAC polymerase can get around that mismatched base. It seems to take a little hit, a couple of cycles difference, but not very much. Certainly not enough to be able to detect an allele that was less than, say 1% of the total number of DNA copies. So IDT came up with a very clever method for addressing this. It's called a blocked cleavable primer. And what it does is it takes a primer, which is here. It's on the left side. You can see it's in black. The green is the template. And in the DNA primer, they introduce one RNA base. So you can see there's a U on the black primer. And they also block the primer. So they put a just some group on the end of the primer to make it so that the polymerase cannot use the primer to prime off of. And then they take advantage of an enzyme called RNase H1. And what RNase H1 does is it cleaves RNA, and it cleaves RNA when there is an RNA DNA complex. So in this case, you can see that 1U that's the RNA, and it's bound to or hybridized to the green DNA template. So you have an RNA DNA hybrid there. And so the RNase H1 recognizes that and it cleaves it. So you can see there on the left side that the blocking moiety has been cleaved off. Whereas if the RNA is not hybridized to the DNA, as you can see below, the cleavage doesn't work and the block remains. There's another picture of it. So now what you can see here is that on the left side, without adding any enzyme, with no RNase, the qPCR creates no product because the primer is blocked. So cycle after cycle after cycle, nothing happens, nothing is produced. When you add RNase, now the primer becomes unblocked and you're able to get product. And so this is thought to be, and as you'll see, it's a much better way, much better at distinguishing between a mutant and a wild type allele. So this is just an example of this experiment. In this case, what they've done is they've run out an experiment where you have on the lower two graphs, you can see that there's a mismatch where they're not adding any. I'm sorry. So on the top two graphs, you have the same mismatch and you have the same slight delay in PCR because it's not setting down right. On the bottom two, you have blocked primers, and so what you see now is you have a delay of about 12 cycles before product starts to get formed. This indicates that the process, unlike the slide here above, where you see that it just goes on and on indefinitely, in actuality, what IDT and what we found is that there, even using these block primers, there's a little bit of leakage, and I don't really know why that leakage occurred, but 12 cycles, that's 2 to the power of 12. That's a number that's, a very large number in terms of the required amount of allele before you actually start to have product. So what we did was we asked them for reagents that they had generated internally and looked to see how much of a delay we had comparing a wild type versus a mutation. And so what we did was we took the mutant and we diluted it. I'm going to just sort of read across. I wish I had my cursor. But we diluted it 1 to 1, 1 to 10, 1 to 100, et cetera, until there was actually no mutant there at all. And what we could see is we were able to demonstrate using these block primers. This was done in duplicate. That's why you have two colors. And I'm sorry that the 1 to 10,000 is yellow, and it's probably very hard to see, but it's there out there with the negative controls. What we were able to determine is that we were able to distinguish a mutant versus a wild type allele out to 1 to 5,000 dilution, and this is pretty good. The 1 to 10,000 is mixed up there with the negative controls, and we weren't able to detect that. So this worked very well for the IDT templates, and we decided we would test our own. So we took some classic oncology mutations, the EGFR, KRAS, ERB2, JAK2, and FGFR2, and we decided we used, again, to find HapMap samples to be able to see whether or not these blocked reagents could distinguish between mutant and wild types. These were defined mutants. We tested both the top and the bottom strand, and the pairs of primers are indicated there in brown and blue. You can see the mutation is being detected by the blocked primer, and then we have a sister primer, which is either there in the blue creating the blue pair, or there in the brown creating the brown pair. So that's why every mutation here, except for the EGFR that has 2 different mutations, as indicated by the RS numbers, every mutation has 4 rows. What we're doing is we are matching both the top. We're using a pair of primers for both the top and the bottom strand. So 1 pair of primers per top or bottom strand mutation. And what we did first of all was simply just to test the primers to see if they worked. And we put the primers on their defined templates. I'm sorry, you can see here that in this little colored table, the 5 rows, the NA numbers, those are HapMap samples. And this defines whether the HapMap sample, and the columns here are the mutations, and whether the HapMap samples were reference, heterozygote, or homozygote. The reds are homozygote, yellow is heterozygote, and green is reference. So we were able to mix these HapMap samples together and to be able to see what the sensitivity was for these various mutants. So first, we didn't do any mixing. We just went ahead and tested how the primers did on in the presence of RNAH1. So these were unblocked primers. And you can see that not all the primers worked, and anyone doing PCRs wouldn't be surprised by that. Then what we did was we did another mixing assay where we mixed the variant together with the reference. And so in this case, on the y-axis, we're seeing the CT. So the lower the CT is, the more product was formed. On the left side of each, and then on the x-axis, you can see each of those is a different mutation. And for each of these groups, on the leftmost bar, the dark blue bar means that we had no variant present at all. So it wouldn't detect anything, and we should get no product. The first thing you notice is that in some cases we did get product, because we would have CTs of, you can see in the ERBB, we had CTs of around 31. So you could slightly get some product, and in cases we didn't see anything. The other thing is that as we go across those colors, we're adding more and more of the mutant. You can see there in red the number of copies of mutant we added. So what we'd want to see is we want to see the CTs dropping. With each addition, we see the CT dropping, at least after, the first dilution is 1 in 20,000. So we didn't really expect to see much there. But after that, 1 in 10,000, 1 in 5,000, we wanted to be able to see that. And in the case of GTAC, we saw a very nice demonstration of the detection of the rare allele. And the ERAB2 is as well. But we didn't see it in all cases. And what we were hoping for is we were hoping to be able to use these blocked primers to be able to create a universal test. And we didn't. And when we were talking about this with IDT, they had ideas about each assay, about how we might be able to increase the productivity. But we didn't want to have each assay be unique. We wanted to be able to do this in the high throughput. So we stepped away from this idea. Okay, so I'm going to continue to talk about rare allele. I'm going to talk about something a little bit more recent for us. And this is using a concept which is actually not particularly recent. We've had unique molecular barcodes for about a decade, but we've applied a tweak to it that's made it rather relatively sensitive. And for those in the audience who are not familiar with unique molecular indexes, I'm going to just briefly describe this. So you can see here in the top blue bars, you can see that some of the fragments contain mutants, which are indicated by the green stars. Some of them don't. And you remember in one of my early images, a question: if that mutant is really rare, is that mutant real, or is that mutant or are we seeing a PCR error or a sequencing error? So what we do is we take our fragmented DNA and we attach a unique barcode to each DNA. And the way this is done is you basically just have 8 or 12 or 16 random nucleotides that are added. So when you take enough random nucleotides, you can get a huge number of different unique sequences. And this is done by, instead of synthesizing an A, a C, or a G, or a T in a certain position, you simply just assign an N. So ACGT is added randomly to the first position. ACGT is added randomly to the second position. And the number of possible oligonucleotides that you can get from that can be calculated by 4 to the power of whatever number of nucleotides you're adding. So, for example, for oligo that's only 2 bases long, you have 16 possible. But 4 bases long, you have 4 to the power of 4. 16 bases long, you have 4 to the power of 16. You can see how this number gets very large very quickly, and you get very unique oligonucleotides. So anyhow, you add these unique oligonucleotides to the fragments, and then you go ahead and amplify them. Now what you'll see is that you get these things called read families. And a read family is all of the progeny from a single unique molecular barcode, a single UMIs. So all those read 1, 2, 3, 4, those all arose from the original fragment. They came from a single fragment. And you can see that every single fragment contains that green mutation, the natural mutation which existed at the beginning of the experiment. And you also have some variants in there which are not common to the read family. And so by using these UMIs, we're able to detect what was there at the beginning or very early on in the experiment. Unfortunately, if a mutation is added very early in the process, it's a little hard to distinguish that from a PCR error. And that is why later on when I'm talking, I'm going to talk about the importance of doing a technical replicate on this. But this is a way of being able to distinguish a PCR error from an actual mutation. So Todd Drewry, who until recently was at Washington University, and he was there when he invented this method, or this variation of the UMI method, created something called error-corrected sequencing. There are a couple of references there that you are welcome to read. And this is sort of the workflow that he did. It's similar to what I talked about with but with some additions. So again, you take genomic DNA and amplify it with basically you're enriching genomic DNA for regions that are of interest. You could be using biotinylated baits. You could be using amplicon sequencing. The second step is you add UMI adapters, those unique molecular barcodes to each amplicon. So it's a very low amplification to start with. Then you add the UMIs, and then you go ahead and amplify up enough so that you're able to create a sequencing library. The libraries are very carefully quantified by digital droplet PCR, so precisely the same amount of library is added into the sequencing. This is one variation that Todd brought in. The reads are aligned, and then you have them grouped into read families, which I was talking about. The sequencing errors that are randomly distributed amongst read families, while the true variants are present in all members of the read family. Another thing that I didn't add on here, and I should have, I'm sorry, both the top and the bottom strand are sequenced. And so this gives you 2 shots at the same fragment. You would need to have that a real mutation appearing twice. And as I mentioned before, you do this using technical duplicates. Using this method, Todd was able to get down to a sensitivity of about 1 in 10,000. He was able to do discovery in 1 in 10,000. Here he's doing an experiment similar to what we were talking about, where he's taking mutant DNA and diluting it into wild type DNA from 1 to 1 down to 1 to 10,000. And you can see that the observation that he made out of the sequencing is comparable to what the theoretical presence of the minor allele should have been. There was one other slide in here I didn't include it. I'm sorry. Just as a little plug for a local St. Louis company, they have the technique that I just described was patented by Washington University, and Canopy Biosciences has exclusive access to this technology. You're welcome to contact them if you'd like to have access to this. They give you, there's a little picture here of the report that comes out. It's very nice. All right, so that's pretty much all I need to talk about in terms of the work that we've been doing for rare allele detection at Washington University. Just in closing, sort of some kind of clinical questions. One is that, you know, we talk about the desirability of becoming more and more sensitive, but at some point, sensitivity ceases to be an issue because, you know, taking a normal tube of blood, you don't necessarily get that many copies of a rare allele in a normal tube of blood. And so the sensitivity should never, doesn't need to exceed the amount of blood that you're taking from a patient. And then there are some biomedical considerations as well. Is that are tumors necessarily DNA leaky? Are medically actionable tumors actually releasing DNA into the circulation? Do we bias ourselves toward the more vulnerable parts of the tumor, whereas the healthier parts of the tumor are not necrosing and therefore not releasing DNA into circulation? And then at what point are detected malignant mutations actually medically actionable? So thank you very much for your time. You have different ways of getting in contact with me. I need to change this last slide. That's my daughter who's just getting ready to go to college. So the picture's a little bit old. That's it, and go Blues.